And this is Unit 4. It's going to have a lot of good information in it. It's going to be an important. There's a lot of concepts to try to make sure that you understand before we get out of here. And this is going to carry on into a lot of other courses, a lot of things such as cell biology, genetics, and so forth. And then 1082. You're going to get a few new terms and things. We will have time when we get done with this, I think, to be able to do a review one day. Now, we have this Friday, we have Monday, and we were doing it the Wednesday before Thanksgiving, so we'll have time. This chapter will go further quick. We'll be able to get done with it pretty much Friday, and then we'll be able to spend some time over everything on Monday for you. This unit is about how your everyday cells, all of your cells in your body, replicate when they need it. Cells don't live forever. They need to be replaced. Skin cells in particular. One thing that you will find, some cells replace themselves and regenerate, others do not. Heart muscle does not, you don't make any more new heart cells. That's one of the things that's so bad about if your heart's damaged, as in a heart attack. Those cells are not going to replace themselves. Neural cells, neurons, will not replace themselves. Some peripheral nerves can repair, but the neurons within the brain cannot, and the spinal cord. Once they're damaged permanently. Cells, as we said, such as your skin, replace themselves constantly. You should have a complete new set of skin every 14 years. By that time, every cell that you've got, if you wait 14 years from now, every cell that you've got in your skin will be gone. Its offspring will be here. It will divide and create new ones. And about cells, also remember this. There are two types of DNA in a cell. The chromosomes that we always talk about are found in the nucleus, and they are called nuclear DNA. There are rings of DNA found in the mitochondria. They are called mitochondrial DNA. Most of the DNA that's associated with mitochondria are there to help you with cellular respiration, metabolism. They control things such as that. You can take any DNA, any cell that you've got will have a complete set of DNA for you. Your skin cells, if you take the DNA out of that nucleus of the skin cell, it's going to have the same genes in it that cells in your pancreas do, or your liver. It's just certain cells will use certain parts of it, but it does have a complete full set. It's no different. So, as we go through this is talking about how those cells divide. And in the next units coming up, you'll get to see exactly how DNA itself replicates. Part is how does the cell replicate? And that is called the cell cycle. The fact that these cells can reproduce is what makes us a living thing, basically. And it is called cell division because in this type of division that we're seeing here, there's no new, basically no new material being put into it. Now remember, this is replicating what you already have. we go through a lot of cells, damage a lot, particularly in the skin, in other areas. And note, it says here that multicellular organisms depend on cell division to develop from the first cell that's fertilized to grow, and as cells become damaged, to repair. Will become damaged over time just from general wear and tear. And this cell division is a big part of this cell cycle, as we will see. This shows you some of the cells and how they will divide. We basically copy the DNA that's there and put one set in one cell, another set of DNA in another. We get two cells. We start with the main cell. From that main cell will come two daughter cells. They're called daughter cells because it's pretty much identical to the one you started with. It's just they regenerate and they become two. Copying what's there. We're not changing anything. This is copying the cells that you already have. We're not changing its DNA. There is a time when we will change the DNA, but that is another process, as you see right here. It's called meiosis, which we will get into shortly here. If you take all of the DNA that's in the nucleus of the cell, that's called an organism's genome. All of the genes. Remember also, genes code for proteins. DNA, most of the time, it's in the nucleus of your cell, and if you were to look at it, it would look like threads that had just been kind of wadded up and put in that little circle. It wouldn't look organized, it would look just a mass. And it has to be exposed inside that cell for enzymes to come along and read the gene, make a copy of it to send out to get a protein made. So it can't be wrapped up. However, before a cell starts to divide, it has to become wrapped in a protein that's going to protect it. And that protein is called chromatin, as we will see. And that is one of the reasons they're called chromosomes. Chromatin is a protein that will wrap them up. All a chromosome is the DNA wrapped in a protein. And that will only occur when cells are getting ready to divide. If it occurred other than that, the DNA, the genes, the code would not be available. So if they're not available, then we could be making the proteins and doing the job that we need for them to do. And that's what this is referred to. It says the DNA molecules are packaged into chromosomes. That comes from that wrapping, the chromatin. If you notice right here, the cell, and notice the nucleus. One of the things that's bad that we do is we misportray what a chromosome actually looks like. There is very few times, only split seconds, where that chromosome is going to be like in an X figure, and then those two are going to separate. But yet we always show chromosomes like that. A chromosome will look more like your finger than anything else most of the times, and you'll get to see that in here. That's what you're seeing on that picture. They're not going to be neatly arranged in little pairs of X's. That does occur, but only at one stage in the cell cycle. And also, we need to get a couple of things straight. That makes it difficult, I think, for people as you go through and try to remember this. A strand of your DNA other words, it's gonna be a copy. Those two strands code for the same thing, it's just a copy. Now, if this is chromosome number 1, you've got one of these that come from the mother, and you've got one that comes from the father. Okay? So you've got two chromosomes. And they very seldom do they look like an X. You'll get to see some pictures of them. What happens when we get ready to make the cell divide? We have to copy both of these. So now we gotta have two of these, because each one is going to go into a different cell. So this one maybe, and this one will go into cell number 1, while this one and this one go into cell number 2. That way we've got a complete set for the two cells. Okay? Oftentimes what you are going to see will be these two written like that as an X. So when you see this X form, this will occur, but only at one stage in the cell cycle, and you'll get to see where that's at. They will pull apart, and one will go into one cell, and one will go into the other cell. And that can be a little bit confusing, because we've got a double-stranded DNA, and now we've got one chromosome from mother and father on each, for each chromosome set. These chromosomes too, you'll find if there is a gene that, let's say, codes for hair color, and that gene is located in this section right there, the same gene will be in the same place on the opposite chromosome from the father. Where they're located is called the locus. We'll get into that, don't try to remember it. I'm just doing this now to explain to you so you can understand when we start to talk about these, what we're actually talking about. When we say a chromosome, we're talking about that, or we're talking about that. When we talk about a chromosome pair, we're talking about both of these. And that would be chromosome number 1. We actually have 46 chromosomes, but there are 23 pairs. In other words, these code for the same thing. The traits are different, the values are different. In other words, the trait could be hair color. The value for hair color can be different. It could be brown, black, whatever. It's still the same trait. Those traits, as we will see, are called alleles. We would say they have a black allele, and that's just that version of the chromosome would be for a black hair color. Yeah.
Speaking of that, along way back when I was learning about the same thing, like in middle school and high school, I learned that actually the red hair gene is the rarest natural human hair color in the world. Do you agree with that?
I don't know. I think fluorescent pink is, and I know there's fluorescent pink because you go down on Vine Street, you can see people with fluorescent pink hair.
That's big.
It sounds reasonable to me. I don't know that it's absolutely, there's one of the things, if you guys take genetics, you're gonna find out.
Mutation.
Well, a lot of the things that you have been told up to this point have been simplified because it's difficult to understand. For example, I just had one lab with ecology people in which we use an example where there's not one gene that controls the trait, there can be two. We can actually have, you can have parents, take Labradors for example. You can have two Labradors, a chocolate one and a brown, have offspring, and from that you can get a chocolate lab, you can get a brown lab, and you can also get yellow because of what is the combination that those genes put out. So it is not as simple as the little 4-box Punnett square. I mean, that's a good theory, and it does tell a lot, but there's more to it than that. A dog's hair coat and color is determined by 46 different genes. Now, you've got a lot of variation that can occur there. It's not saying what we've not taught you is not being true, it has, but it's been very generalized, and we will muddy those waters up a little bit later, because we will get into why some of these things that can happen, exceptions. But those are chromosomes, so remember these chromosomes right there. And try to, you've got one from your mother and one from your father, set one. You'll have one from your mother, one from your father, and chromosome number 2. We have got 23 sets of chromosomes. Half the set comes from your mother, half the set comes from your father. And that's pretty much getting into the heredity we'll talk about next chapter. Remember, chromosomes are covered in a protein called chromatin. It's actually more than just a protein, but the main part of it's protein. Notice it says it condenses. That chromatin will wrap around, and it will take like you just thought of a DNA as a piece of wire. And if you've ever seen a pipe cleaner, it's got all that little fuzzy stuff on the wire. That's what a chromosome looks like. It protects that DNA in the center. There are only 2 kinds of cells in the body. Now, we can divide cells up into many, many different groups. There's many ways. But the main base 2 types of cells are called somatic and gametes. And they're real easy to figure out. Gametes are either the egg or the sperm, depending on male or female. Every other cell in your body is called somatic. So it's pretty easy to remember. To remember gametes or egg, sperm, everything else then is somatic.
What I also remember is gametes are the only cells that undergo meiosis.
Yep.
While the somatic cells and everything else undergoes mitosis.
Well, meiosis actually starts out with a mitotic cell, but the process, that's the way it divides.
Yeah. It's part of the cell cycle.
So remember those 2. The reproductive cells are gametes. Everything else is called a somatic cell. Whether it's a skin cell, a muscle cell, doesn't matter. Now, remember we've got DNA sitting in the nucleus of the cell, and it's starting to get ready now to divide. Notice DNA is replicated and the chromosomes condense in getting ready for this. Now there's a whole lot said right there that we skip over. The DNA is replicated. It will be replicated in a particular phase that we'll talk about, but we copy it. The chromosomes condense together. Now we can't copy the DNA if it's in a wrapped up chromosome, can we? We have to know what the code is. So the DNA has to duplicate before it becomes a chromosome. And then the chromosomes can form and then get organized. Each duplicated chromosome has 2 sister chromatids, joined copies of the original chromosome. That's the chromosome. Each one of these would be 2 chromatids. Copy. They are going to be joined together. Remember I said there was a particular time where some of those look like an X? That is because these 2 will join together in an area down here, and it's joined by a constriction called a centromere. Now this shows you, if you look, each of these is a copy of your original chromosome. This is the original, and then you've got two fuzzy copies now. They're fuzzy now because they've got the rectin chromatin. They will be joined right here. Remember, you have to copy those. Okay? And as you will see later, these strands will pull apart. You'll make one copy from that strand and another copy from the other strand is where you get the two different ones. And in that process, they will be joined together. Where they are joined together, that constriction is called the centromere. And because each of these came from the same chromosome, they are called sister chromatids. During cell division, the two sister chromatids of each duplicated chromosome separate and move into the nuclei of the cells. That's what's going to happen. Once they separate, these chromatids are now called chromosomes. We've got them here separate their chromosomes. So this starts us from scratch. Notice here, we've got a chromosome. Now, they draw these as that structure looks like your finger or earthworm or something. They don't draw them showing you the double helix of DNA and how there's two strands. They're just showing you the whole DNA strand. This is what it looks like by itself, and this is what it looks like as a chromosome, because now, the reason they don't draw it this way, this is being covered by that chromatin, that protein, for protection. Chromosome duplication, including DNA replication and condensation. So we come with one chromosome. This is going to be happening basically before it gets wrapped in the fuzzy protein. We'll make the copies, as you see over on the right. And then we've got two of these now that are joined together. We went from one to two copies. That's where we get the X phase from. And then they will separate and go to different cells. So this chromosome looks like that, this one like that. They're still DNA, they're just wrapped. We think of them as one, but you've got to remember, you've got the double helix inside of each one.
Okay, so I was always confused. I thought that the actual X was called the chromosome. So that's actually, each one of them is a chromosome, and then when it's in the X form, those are the sister chromatids still, right? Because they're
chromatids whenever they're joined.
When they're joined, okay.
When they separate, they become chromosomes.
So there's not a specific name for the joined pair, right? I mean, you just call them sister chromatids.
Sister chromatids.
Okay.
And that happens only in one spot, and it doesn't last very long. This is the one that I was telling you about. We always get people calling them X chromosome. They're in there for a very, very short time. They look more. This is the way a chromosome looks, but it's top, not that X. The X is when it's got its copied partner there.
Thank you.
But notice over here on the right, it's still DNA. It's just wrapped in a protein. Now, when the cell division gets ready, we've got it. It happens in parts. We have to have a place in the beginning up here where the DNA is actually going to be copied before anything else can occur. Then we will get the name for that particular stage. After it gets copied, then it goes through a process called mitosis, which is the division. We've copied the genetic material. Now we are going to divide it into two separate cells. That process is called mitosis. After we put the chromosomes in separate cells, we now have to complete the separation of those two cells. That is called cytokinesis. And as we've already mentioned, the gametes will be produced by meiosis. Some people call it meiosis, whatever. Doesn't make a difference, but it's mitosis does somatic cells only. Meiosis does the gametes. It is a different cycle. We will get into it next chapter. Notice, here's one big difference between meiosis and mitosis. In mitosis, it's basically cell cloning. They're the same. Meiosis produces cells that are not identical genetically. They will be different, which is why none of you look exactly 50% like your mother and 50% like your father. Don't work that way. What happens is they blend, basically, the traits, and you become unique because you've got a blend of their two traits. You may see resemblances in certain things, but it's a blend. Here are the phases of the cell cycle. Now, this one, interphase, should come first. I don't know, have no clue why they put it second. It should be first. And interphase is where, as you see, the chromosomes will be copied. And also in this phase, we're going to need two cells now. We're going to have to grow two new cells. So all of the little organelles, all of the enzymes and the proteins that have to be made, that kicks in. It starts to make the membrane for the two new cells. It will make this inner stuff that makes up the cell, organelles and whatnot. It will make all of that during interphase and have it ready. In mitosis, once those cells are ready, once the chromosomes have been duplicated, once their DNA and then the chromosomes formed by wrapping it in chromatin, then we can start separating the cells and putting them into the two different cells. That is mitosis. Oftentimes, mitosis and cytokinesis are called just the mitotic phase. They're under the mitotic phase. But they are two processes. Mitotic phase means the whole thing, mitosis and cytokinesis. Now this first phase of where cells begin to divide, notice 90% of the time is going to be in, of that whole cell cycle, 90% is going to be in the preparation stage, getting it ready. They are called G1, S, and G2. Now the reason they're called these phases is because there are checkpoints along the way. And once the cell starts to divide, it has to get through that first gap, that G1 checkpoint. If it cannot get through that, then the body will destroy those cells. If something's wrong and it's not working correctly, you've not made a good cell, that will stop there. The S phase is called the synthesis. This is where we copy DNA and get these cell parts ready. And the G2 phase is basically just once we get the cells started, continued growth until they get to the point where they are ready to become a new cell on their own. And notice at the bottom, it says the cell is always growing. It's changing inside. You're getting everything ready to make two new cells. all through all of these, but only in the S phase, which is called the synthesis phase, are the chromosomes duplicated. Now still, it should be not chromosomes duplicated, but DNA's duplicated, it becomes chromosomes. And this shows pretty much what's happening. If you start here, G1. This is how much of the time is going to be spent in that G1 phase. That's getting new cell parts ready to be made, organelle, membranes, and all of that that's occurring. The S phase, notice here, it's DNA synthesis. That's when we're copying the DNA and are going to turn it into chromosomes. The G2 phase is kind of the cells are going to complete, they're going to mature, get to where they're ready. So we've got 2 cells, and then mitosis starts. Mitosis will be pulling those chromosome sets apart, so they go into those 2 newly made cells. Cytokinesis will be those 2 newly made cells which are combined will then cut and separate. Out of the end comes these 2 new cells now. Big thing to remember, S phase, that's where DNA is copied. Mitosis is where the copied DNA goes into separate cells. Mitosis is divided into 5 phases. Remember, this is the part where the DNA has been copied. Now we're going to fold them into 2 different forming cells. Those cells are not completely formed yet. Think of them like as an egg that is connected. If you were to break 2 eggs in a pan and you've got the yolks representing the nucleus, which is what it is, you gotta know that. An egg cell is just, or an egg is just a giant cell. That's all it is. And then you got the yolk. And then that's the way these cells look. They've not separated, come apart. They're growing and going to come apart, but they're not there yet. These are the 5 phases you do need to know. There's a lot of things that happen in these phases. I will narrow it down for you a great deal. Prophase, prometaphase, metaphase, anaphase, and telophase. I'll tell you what they do in a minute. We'll look at visually, you'll get to see it. One of the things that there is no distinct point. When does telophase end and cytokinesis start? That is kind of variable. When cytokinesis starts, telophase is still going on. So there's no place where telekinesis stops, or telophase stops and cytokinesis begins. These are the things that you need to be able to see. And notice what's happening here. G2, prophase, prometaphase. So this is what it looks like at the end of interphase, right here. Notice how many chromosomes are all packed into that one nucleus because they've been doubled. Notice at the top, these 2 little guys right here, centrosomes, not centromere that we looked at, centrosomes. Notice what's happening. During prophase, these 2 centrioles, they're called, it's a centrosome, but each one is called a centriole, they're pairs. One goes to one pole, one moves down towards the other pole. They are moving. Also notice, in prophase, the first phase now of mitosis. Chromosomes start to organize. In other words, they're getting matched up with the pair they're supposed to be matched up with. Believe it or not, sometimes that goes wrong. Sometimes you can get conditions in which these chromosomes don't line up, and it causes certain defects that you'll see if you take genetics. So in prophase, centrioles are moving, the chromosomes are together with their sister chromatids, becoming more structured. Then, prometaphase. Notice they are starting to separate. Prophase, they become more organized. Prometaphase, they are starting to separate. Look at those yellow centrioles now. They are on each pole of the cell. And notice how each one of these are called kinetochores, which are microtubule, or you can call them spindle fibers, which is what most people call them. These attach to a chromosome, sister chromatid there, this one to a sister chromatid there. They are going to start to shorten those fibers, and then they will pull the chromosomes apart. Prometaphase. In prometaphase, you can put basically spindle fibers form and start to separate the chromosome or the chromatids. After pro-metaphase, that means before the middle phase. Look at metaphase. Those chromosomes line up on the horizontal equator of the cell. That thing there that says metaphase plate is going to become the dividing line between the two cells. Now, we've got one big cell we've been dealing with that's going to become two cells. This will become one, that will become one. Notice that's metaphase. In anaphase, they have just started to pull apart. You've got these fibers that are now shortening, and they're going to pull them towards that other pole. By the time telophase comes, as you can see, these are going to continue to be pulled towards the pole. Then telophase will begin. Telophase, you know, if you go into the kitchen, for example, and you've got meat cleavers, cleave means to cut. So that is a cleavage furrow that starts, which means it's starting to cut, pinch itself off. It's going to keep getting smaller and smaller, less attachment until they separate. That process is cytokinesis. And notice something else that happens in telophase. The nucleus starts to form. So notice now we've got these chromosome copies pulled to the pole and a nucleus, a nuclear membrane envelope is forming around them.
Out of curiosity, did you guys already do the mitosis lab in your lab? Okay, because I did it, so I kind of got to see firsthand under a microscope on
We have
real
not done
cells
it yet.
how this looks.
We have not done it yet.
And
We're
I
getting
actually
ready
got
to.
some pictures too.
So that process is mitosis. At the end, the two cells will completely come apart, make two new cells, and that is cytokinesis. So let's look again at what's happening. Interphase. That's where things are copied. Everything that's going to be needed to make two cells will be made here. The DNA will be copied in the S phase, synthesis phase. And all of these things will continue to grow and mature through the G2 phase. Then we move into mitosis. That is where we're going to take the material that's got enough for two cells and two sets of chromosomes, start to organize and pull them together. And notice, this is the last phase. We're coming out of interphase right here. In prophase, I guess I should go up once again. In prophase, we're starting to see organization, sister chromatids. In prometaphase, spindles have attached to the sister chromatids and start to organize them in that horizontal plane. In metaphase, they are lined up. The mitotic spindles are in place and now start to pull each of those sister chromatids apart, pulling them one set towards one pole. Anaphase, as they continue towards the poles, at the end of anaphase, they will start to make, you notice here, this cell has elongated, they don't depict it as round now, it's starting to pull. It will eventually have this cleavage furrow, and then during cytokinesis pull apart. Nuclear envelope will start in telophase. Chromosomes will be encased now within the nucleus, and we've got two cells that have formed. Some things to remember. How many times did the cell divide? One time. And how many cells did we get? Two. This will change when we talk about meiosis, because it will divide twice and wind up with four cells. In mitosis, one cell division and two cells. And remember, they now have a full, complete set of chromosomes. They are called diploid. OID is going to refer to how many sets of chromosomes do you have. If you have only half of the set, like a gamete will, they're called haploid. Their number, chromosome number, is N. These cells that divide and make a whole new functioning cell, somatic cell, have a full set. What we're looking at here, they have a full set of chromosomes. They are going to be said to be diploid, diplo meaning two. Some plants can be triploid, have three sets. And I want to make sure that you understand here what's going on with this. Anytime that you are asked, and this happens every year, you still don't listen. If you are asked what is somebody's chromosome number, they're wanting to know, is it N, is it 2N, or 3N? They don't, if I ask you what is a human's chromosome number, you don't say 46. That's how many chromosomes it has. Its chromosome number is diploid, 2N. If we take one cell, 46 chromosomes, we copy them, we wind up with 2 cells with 46 chromosomes. Right? That is a mitotic cell. If we don't copy the cells anymore, but we divide them, now we've got 3 cells, or 4 cells, that are gametes. So mitosis would be going to this part and stopping, and just copying the cell. And if we continue down here, this would be meiosis, which is what we'll do next. Meiosis, as you'll see, will have 2 parts to it. Meiosis 1, which is basically going to be this. And then meiosis 2, which is this. It's not as complex as it sounds. Take a mitotic cell, don't copy any of the chromosomes, and just take the full set it's got, put half in each. Now, let's look at you. If this is you, remember, you got 46 chromosomes, 23 from the mother, 23 from your father. Okay, when this cell divides, what we got here, if we copy these like this, and then we make 4 cells out of these, that means you are going to have 2 chromosomes for that set. You're going to have 2 possibilities from your mother. You're going to have 2 from your father. So, when you go through and you make eggs, those eggs, that's your possibility. Doesn't matter if it's eggs or sperm, whatever, it's, you're carrying those chromosomes from both mother and father. That's the combination you get. And when does a female mammal, humans, when do the eggs form? Actually, before birth. Believe it or not, the eggs are there. What happens is, they start to go through this, but stop. They don't complete it. They just get started. And then, throughout the life of the female, that's those eggs, it will start up again and complete. That's one of the things that is thought that if you look at Down syndrome, that it's been noted that that is higher likely in older women. And they think it's because the eggs have just been around so long, it's been so long between the process starting and stopping and doing this, that's where things could be going wrong. I don't know that for sure. But these develop, and just keep in mind, you're going to put out 4 eggs or sperm, and each with a different allele. Now, how do you know which one of these you're going to get? Totally random. That's what independent assortment is, as we will look at in a minute. It just means you get whichever one you get. Nothing grows. So see you Friday.